All right. All right. So hello, everyone. This is Mohamad Harastani. I have a PhD in bioimage informatics. I'm currently a postdoc in the IGBMC. In there, we do genetics, molecular and cell biology. I've done my PhD in between Sorbonne University and Université Paris-Saclay in Paris, France. And also during my PhD, I was collaborating with the IGBMC, and finally they offered me a position, and here I am. So my PhD was about conformational variability analysis of biomolecules in cryogenic electron tomography. So let's start. Okay. So biomolecules change their shapes or conformations while they achieve their biological functions. If you look at the one on the top left, you see it bending left, right, and front and back. This is while it achieves a function in DNA replication. When we describe this motion, we say that it is continuous, meaning that it does not exist exclusively at left, right, front, back conformations, but at many conformations in between. Opposite to continuous, we have discrete type of variabilities, such as a molecule binding or unbinding a substrate. And we can have combined discrete and continuous type of variabilities, such as this elongation cycle of the ribosome, where it binds and unbinds different ligands while continuously changing its shape. Of course, understanding the shape is important to understanding the function, and understanding this shape variability becomes even more important to understand how to develop drugs or maybe fight cancer. In order to look at biomolecules and their conformations, the tool that we usually use is cryo-electron microscopy, because it uses frozen hydrated biological samples that preserves the native structures. And there are two methods that can be used. The first is 2D image analysis method called single particle analysis, and the other is called cryo-electron tomography. And I will explain these two in future slides. In my presentation, in my thesis, I was working on computational methods for analyzing continuous conformational variabilities in cryo-electron tomography. These two methods are called HEMNA3D and TomoFlow. And in this presentation, I will show you these methods. And I'll show you how we tested these methods on synthetic data and then applied it to an experimental case study for nucleosomes in cells in situ. The first method is, and the most common, is called cryo-electron microscopy single particle analysis. In this method, you would start from a sample that has already purified molecule or protein that you would like to visualize in 3D, or you would like to get its shape. It could be, for example, the coronavirus, and this is how actually it was obtained, the 3D structure of the coronavirus. You would start from the sample, you would take a droplet, you would place it on what we call an EM grid. It's a grid that has tiny holes in the micrometer radius. And you would place your droplet that has many copies of your molecule, and you would freeze it rapidly by plunging it in liquid ethane and liquid nitrogen. Then what you form is a thin layer of vitreous ice, as you see on the bottom right, that traps your macromolecule in different orientations. Then you would image this under the electron microscope, and you get what we call a micrograph. In this micrograph, there are many copies of this protein. You would pick them, and then through sorting their orientations, you can have a 3D reconstruction of the protein in your sample. A single particle image data set can contain hundreds of thousands, tens of thousands to millions of different copies of the protein. This is because of the automation in the data acquisition. But they come at different orientations and possibly different conformations, and in very challenging noise. The noise comes because biological samples are very fragile, and electrons damage the structure. They make everything blurry. So you need to use low electron dose in order to preserve the structures. So what we need to do is to sort out the orientations and come up with 3D reconstructions. But if the sample is heterogeneous, let's say you have flexibility hidden in the data and that you would like to understand, then your goal becomes to reconstruct as many 3D reconstructions as necessary in order to reveal this conformational variability and to understand the flexibility of your protein. Methods that deals with this conformational variability are usually of two types. The first is addresses this variability as discrete, even if it is continuous, and tries to address it with classification. And the second is for continuous conformational variability, where it tries to map the conformations to what we call the conformational landscape or conformational space, and tries to, by modeling this conformation, tries to understand the flexibility. And from these methods, there will be the methods that I developed later on, and they will be explained in detail. Now, cryo-electron tomography is the second method. It is honestly more interesting, at least to me. And you would start by not purifying proteins, which cannot be done for every sample, for every protein, but actually vitrifying cells or tissues, freezing cells or tissues, and then sectioning them with one of two methods that are common: cryosectioning, which is like mechanically sectioning the cells with a diamond little knife using a machine called a microtome, or with cryofib milling, which is like milling the region, thinning the cells by shooting focused ion beam on the sample. In any case, you would get a thin section that of your cell that is placed on an EM grid. Again, you would put it under the microscope, but this time, because the cell environment is very overlapping and complex, a single 2D image might not be enough. So you usually tilt the sample under the microscope, usually from -60 to 60 degrees, because of the physical limits for tilting above or below +60 and -60. And you would get what we call a tilt series, a series of micrographs, one for every tilt angle. And then you would reconstruct this tilt series to 3D. And you get what we call a tomogram. In this tomogram, you have, again, similar to the micrograph, different copies of the macromolecule at different orientations and possibly different conformations. You would pick them, you locate them in 3D, and through data processing, you get the 3D structure of the molecule that you are addressing. Now, tomographic reconstruction in tomography, cryo-electron tomography, can be explained based on the Fourier central slice theorem. And this theorem tells you that a 2D projection of a 3D object, the Fourier transform of this 2D projection corresponds to an actual slice in the 3D Fourier transform of the original 3D object. And if you try to see what happens in tomographic reconstructions, since we are usually limited in the tilting axis by -60 to 60 degrees, the Fourier 3D Fourier transform of the tomogram looks like that. It has a missing wedge region in the Fourier space, corresponding to the tilts that are above 60 or below -60. And these cause some artifacts, reconstruction artifacts called missing wedge artifacts, making the tomograms not to be a loyal representation of the signal that actually exists in the sample. Meaning this will make tomography data even more difficult to process. right? Now, in order to increase the signal and correct for missing wedge artifacts, what usually we do is rigid body alignment and classification. Now, this rigid body alignment is called subtomogram averaging. We start from a tomogram that we have already picked the positions of the macromolecule that we are analyzing. These positions contain the molecule at different orientations and, of course, possibly different conformations. We start by just summing these copies to an average, and we use this average as a reference for alignment, for rigid body alignment. And then we find the new sum of the aligned subtomograms. In this iterative process, after a few iterations, we get the global subtomogram average and we get aligned subtomograms. And those can be used for classification. This is a demo. So I'm starting from a molecule, an atomic structure of a molecule. It is just it has no biological importance in this sense, but it's just like a toy example. I'm simulating two movements. I use for this simulation a method called normal mode analysis that I will be explaining later. And then I'm combining these two movements more or less in a linear fashion to generate a combination of volumes that have different conformations. So in some of these volumes, if you see the green middle one, you see a more closed structure. And some of them have more open structure, and I have everything in between. And then I add noise, a little bit of noise, some other microscopy, what do we call that? Modulation functions called the CTF, contrast transfer function, rotating and shifting the molecules, and then applying missing wedge artifacts to create what we call synthetic subtomogram. And then I applied subtomogram averaging, this iterative process of getting the global shape that is in the data. After 10 iterations, I have a decent average, and I can have aligned subtomograms. But now the question is, where did all the movement that I've hidden in the data go? And how to extract back this movement? Because yes, we have the average and we have aligned subtomograms, but where are the movements? So this is what I was actually trying to address in my thesis. And I've developed two methods that I'm going to show you. The first is called HEMNA 3D, based on normal mode analysis. Normal mode analysis is a method to simulate molecular mechanics that is usually used to fit what we call an atomic structure. Those atomic structure that usually we see on TV, they usually come from another method called X-ray crystallography. To fit this atomic structure to another map that usually comes from electron microscopy, and to get what we call an atomic model of this map. And the second method is called tomoflow, and it is based on optical flow. I think as computer scientists, you will understand this one. So let's start with the first method, and normal mode analysis. So normal mode analysis, as I said, it's just a method to simulate molecular mechanics for usually atomic structures. And it is based on breaking down the different movements that an atomic structure can take to low frequency collective movements all the way to high frequency localized movements. And what the benefit usually of this normal mode analysis, if you have an atomic structure that has n number of atoms, it will have 3n degrees of freedom. And usually the low frequency collective movements, just like if you have any transform, Fourier transform, wavelet decomposition, some of the collection of the low frequency collective movements is enough to give you a good representation of, in this case, the possible movements a molecule can take. Now, usually you would use an atomic structure to get normal mode analysis, but if you don't have that and you have an EM map or a subtomogram average, the one that I've shown before, you can represent it with some 3D Gaussian functions. We call them pseudoatoms, and you can also find its normal mode analysis or its normal modes. And finally, an example, if you have an input structure, the different normal modes, you can have many different normal modes, but the subset of the first 20 might be enough to explain how this molecule might move. And everything above 20 might be too detailed movements that are not very essential to understanding how this molecule essentially moves. Normal mode analysis itself is actually very simple. You would represent the connection between any two neighboring atoms with an elastic spring, and this helps you to get a very simple potential energy function of the whole system. You would use some harmonic approximation of this potential energy around the given state as energy minimum, and you will get through this harmonic approximation a Hessian matrix, or matrix of second derivatives. The eigenvectors of the Hessian matrix are normal modes, and the eigenvalues are frequency squared. So it now order the normal modes by their frequencies, and you will get a subset of the first few normal modes, let's say 10, 20, and those will be enough for you to analyze the conformations, let's say, of a molecule. So the main application of normal mode analysis is normal mode fitting. And I've said that before, but given an atomic structure, the one that you see in blue on the left, and an EM map, the one you see in gray, the goal of normal mode fitting is to get an atomic model of the EM map by fitting this atomic structure inside the EM map. You would calculate normal modes, and then normal mode fitting is this process of iteratively trying to fit the structure by searching for normal mode amplitudes or combination of normal modes that actually, by applying them to the atomic structure conforms to the EM map. And those normal mode amplitudes, or the Qs, is the output of the fitting, and of course, the fitted model. Now, you can fit, of course, an atomic structure to an EM map, but again, you can fit an EM map to an EM map. It's not usually useful in general, unless you have such an application similar to the one that I'm showing in my thesis, which is using a subtomogram average as a starting reference and fitting them to those subtomograms. And this is what we have actually done in HEMNA3D. So HEMNA3D takes as input subtomograms and tries to map them, or it maps them through this normal mode analysis and normal mode fitting to what we call a conformational space. In this space, every point here represents a subtomogram, and close points represent subtomograms that have similar conformations. Well, once we have this space, we can do two things. The first is to group subtomograms that correspond to points in dense region in this conformational space and have their corresponding subtomogram average that shows different conformations. And the second is to fit lines that follow manifolds along the densest region in the data distribution in this conformational space and generate movie-like animations that might reveal trajectories of conformational variabilities. The way we've done it is to extend normal mode fitting to work for subtomograms, and that is to include both missing wedge corrections during the fitting, or missing wedge compensation during the fitting, and to search for rigid body alignments, because the rigid body the conformation, the orientation of every subtomogram is also missing, is hidden. So we do at the same time rigid body search and conformational variability search, taking into account that there are missing wedge artifacts. And the output of the fitting of every subtomogram are those Qs, or amplitudes of normal modes. And those amplitudes of normal modes finally can be mapped to a lower dimensional space, for example, using PCA, to get the conformational space where we can generate subtomogram averages of different conformations and animate trajectories. To test HEMNA 3D, we generated many data sets. This is one of them. By deforming the structure, orienting it, and shifting it, and then applying some missing wedge artifacts from -60 to 60. And of course, some microscopy features or modulation functions. Then the ground truth that we were seeking is a linear combination between two normal modes, and we use 3 normal modes to try to retrieve these hidden amplitudes, and we were able to get them back. Now, the second method that I'll present is called TomoFlow, and it is based on optical flow. So optical flow is a method that can take, that usually takes 2 images as input and tries to find pixel-to-pixel correspondence that will map one image to the other. So usually these 2 images, they come from 2 consecutive video frames, like the images of this Rubik's Cube, where it is actually rotating. And the optical flow, the one underneath, or the one in the middle here, shows where did each pixel go from one image to another. Now, the early concepts that helped developing this optical flow in the 1980s were based on 2 assumptions. The first is that the brightness is constant. Then when a pixel translates between one image and another, then it preserves the same brightness value. And the second is that the motion is small, that the pixel does not travel more than 1 pixel in range. But of course, those limited the optical flow capabilities to just very small motions, and to have to be very sensitive to brightness changes. In kind of the new era of optical flow after 2003, optical flow calculation, there's farm-back optical flow that is first based on finding the translation of every pixel, finding the flow by taking into account its neighborhood. This is based on quadratic expansion of every neighborhood before finding the pixel displacements. And this reduced the brightness errors and sensitivity to noise. And the second one is to estimate bigger movements. Farm-back optical flow estimates the optical flow at multiple scales. It starts from the lowest scale, where movements can be narrow, and it upsamples it and refines it until it reaches to the original scale. And of course, this helped to estimate bigger movements. What we have done, we used optical flow for 2 purposes: to refine rigid body alignment and to do conformational variability analysis. And I'm going to show how we've done that. So we start from V is a reference, and S is here a subtomogram. So V could be this global subtomogram average or some external reference of the macromolecule that we are trying to analyze. What we do is we finding some difficulties too. We find, so these 2, we assume that they are at different conformations, but S is actually noisy and is kind of buried in noise. We find the optical flow between these 2 volumes in the presence of some Mascov region of interest. And we find the optical flow, and then we apply it to V in order to get an estimation of S, or estimation of the conformation of S, that is actually similar to S, that will reveal its conformational change or the hidden conformation in it. Then we designed TomoFlow that takes as input some missing wedge corrected and aligned subtomograms. And these are, of course, not straightforward to obtain. I will show in the next slide how we get to this input. But the main idea is once we get these missing wedge aligned and missing wedge corrected and aligned subtomograms, we use the subtomogram average as a reference in order to find the optical flow for every subtomogram in the presence of some mask of region of interest. We map those subtomograms to a matrix called gram matrix, a matrix of pairwise inner products, and we apply PCA to it. And we get this conformational space. And again, from this conformational space, we can group some dense regions and get subtomogram averages at different conformations. And we can generate movie-like animations by displacing the average along some data distribution manifolds and showing some trajectories of the molecule that we are analyzing. So the pre-processing steps that we followed in order to get those missing wedge aligned and missing wedge corrected and aligned subtomograms starts by conventional subtomogram averaging, similar to the one that I've shown in my demo in my few first slides, followed by filling the missing wedge in Fourier space with the region that corresponds in the average. So we get the global subtomogram average, we align it to every subtomogram, and then we take from it, or we borrow, the missing wedge region in every subtomogram and add it to these subtomograms. And then we do the inverse Fourier transform. Once we have those missing wedge filled subtomograms, we pre-align them based on the information in the subtomogram averaging. We do the optical flow matching that I showed a couple of slides ago. And we do subtomogram alignment again in order to refine or correct for any missing or residual rigid body alignment errors that existed in the data from the traditional subtomogram averaging. Once we have this table of rigid body refinement, we can combine it with the original rigid body alignment and get a refined subtomogram average and refined aligned subtomograms. This is an iterative process that usually we repeat a couple of times to get rid of any residual misalignments, rigid body misalignments, and to correct for missing wedge artifacts, partially correct for missing wedge artifacts. So we tested TomoFlow on synthetic data sets. This data set that I'm showing here was generated from a very common method called molecular dynamics simulations that shows again a continuum of transitions for a toy macromolecule that goes from an open state all the way to a closed state. And the data set had all the combinations in between. So of course, this data set was synthesized as synthetic subtomograms by adding gradually different levels of noise until it is really like nothing is visible in the data, and missing wedge artifacts. As you see, there's some missing wedge region in the Fourier space, and those missing wedge artifacts, you can see elongations even when there's no noise, and some what we call gray artifacts, data anisotropies. Tests show that the rigid body refinement of TomoFlow was always able to fix for some of the rigid body misalignments by reducing the angular and shifting errors. But also for the highest noise value that we tested, we were still able to retrieve back some subtomogram averages that reveal the hidden conformational trajectory in the data. So now from this slide on, I'm showing how we've applied both methods to a data set, an experimental data set for nucleosomes in cells, and how we actually revealed some of the hidden conformational trajectories of the nucleosomes in cells. So nucleosomes are actually what make chromatin or chromosomes in eukaryotic cells. The DNA of eukaryotic cells is packed into chromatin, and the chromatin is an array of nucleosomes. As you see here on the top right, it looks like a bead on a string, and the bead is this nucleosome. So there are, of course, many nucleosomes packed together inside the nucleus. In the figure on the left, figure A, you see a region of chromatin. Beside it, you see the nuclear double membrane, and outside the cytoplasm. If you zoom into this region, you find nucleosomes of different orientations. So this is a image on the left is a 5 nanometer thick tomographic slide. Now I will show you how this data was obtained. This data is coming from a brain of a Drosophila embryo. We start from culturing the embryos, stealing them from their flies, and then isolating them, freezing them using a machine called high pressure freezing that increases the pressure and lowers the temperature at the same time. Then sectioning them using a microtome with a method called cryosectioning. And those sections are placed in the microscope in order to obtain a tomogram or a tilt series that is finally reconstructed into a tomogram. So the tilt series you see here on the left. So these are images that are from -60 to 60. Even in the tilt series, you can see the double membrane in some of the images. And inside, there's the chromatin region. Here you see this tomogram. It went too fast. Nucleosomes, those are ribosomes, and there's the double membrane of the nucleus. So we picked different nucleosomes, 650 of them. We had the global subtomogram average, the one that is actually shown here in red. Beside it, there is the atomic structure that is coming from crystallographic studies. HEMNA-3D, applying HEMNA-3D to this data set using 6 low-frequency normal modes, which is 6 is just an arbitrary number. We could have used 10. Resulted in this conformational space. We located in this space 4 densely populated regions. We found the corresponding subtomogram averages, and it showed different conformations. We also placed some lines, fitted some lines that follow the densest region in this space. And we showed 2 movie-like animations of a nucleosome that is actually changing conformation. These 2 conformations are called the gaping, this one, or clapping movement of the nucleosome, and the breathing movement. Okay. Now we also applied to the same data set TomoFlow. We used the global subtomogram average as a reference, and we did different iterations of missing wedge filling or correction and rigid body refinement. And we also got a conformational space that we were able to show different conformations. They were not as big as the previous ones, and I will discuss that in my next slide. And we also showed a movie-like animation of the nucleosome that is also kind of simultaneously clapping in a very small amplitude here in the second figure, second line, it is obvious, and breathing. In the first line, it's also somehow obvious that the 2 DNA gyres or arms of the nucleosome are going towards each other and away from each other. So as a summary, I presented 2 methods, HEMNA-3D and TomoFlow. Now, HEMNA-3D requires a selection of a subset of normal modes. And if this selection is very small, if the set of normal modes that are used are very small, then you risk to have bias. And if this subset of normal modes is very big, then you start to overfit noise and local movements that might not be necessarily true. TomoFlow has also some pros and cons. So it has a trade-off between sensitivity to noise and extracting large movements. So TomoFlow is more conservative and it extracts the movements from the data. And it needs like the consensus of the data in order to extract you a movement. And of course, both have parameters. TomoFlow also have the parameter of what will be the region that you would like to use in this quadratic expansion during the far and back optical flow calculation. And if you use very small region, then you will be able to extract large movements, but it will be sensitive to noise and it will overfit. And if you put this region to be big, very big, the flow will be almost zero and you will not see any movements. So both have these kind of trade-offs. So in my perspectives, I would like to combine both methods. I already have a clear idea how to do that. We found a way to project normal mode amplitudes, optical flow amplitudes, optical flow motions on normal modes and get some amplitudes. And the two methods can be combined. But of course, this comes on the expense of the computational complexity and the time of execution. And of course, to develop other methods that could be inspired from other existing methods in 2D. What I have not said is that there are this continuous conformational variability research is very popular in cryo-electron microscopy. But the things, due to the challenges in the data, like missing wedge artifacts and the very low contrast, still have not attracted many people to try to attempt to extract these movements for cryo-electron tomography. So there are some different techniques that are based mainly on deep learning that can be used in this problem. So finally, I'd like to thank my collaborators, my supervisor, Slavica Jonic in Sorbonne University and her team, my co-supervisor, Amélie Le Forestier and her team, and my current PI, Mikhail Eltsov and his team. The people in Spain, those two methods that I have shown, they are implemented in an open source software called Continuous Flex as a part of another open source software called Scipion. And I'd like to thank the Scipion team for their support in how we have actually implemented these methods. And I would like, of course, to thank my family, friends, and colleagues. And thank you also for your attention, and I'll be happy to see if you have any questions.
Thank you for this nice presentation.
Thank you.
As I'm not working currently on this topic, it might be a little bit basic questions. I want to ask about model performance metrics that you used for your models. Are there only distance and averaging?
Well, yes. Well, the methods cannot be tested easily, let's say. The performance of these methods, let's say, tested on synthetic data, since you have some ground truth, can only be done by seeing how distanced are you from the original synthesized, let's say, movements or rigid body variabilities. So to see how close we are to the movements, there are some metrics that are very similar to the mean square error, but in terms of these 3D atomic structures, they're called root mean square deviation, RMSD. And we have shown somewhere in these papers that, at least for HEMNA 3D, that we are about half an angstrom on average away from the actual conformation. So an angstrom is 10 to the power of minus 10 meters, and atomic resolutions usually come from 3 angstrom, which means that half an angstrom is a quite low average for the error. For rigid body variabilities, you would usually know what is the, in synthetic data at least, you would usually know what will be the ground truth rigid body variabilities, and just the mean and the standard deviation are usually enough to express how distanced you were to a line model or your reference to every subtomogram or every image. So yeah, I hope this answers something.
Yes, thank you.
Welcome.
Are there any other questions?
So maybe I can ask. So thank you for the presentation. That was a very new topic to me and a very interesting one. So maybe I can ask a question, but that maybe be very basic or a different question.
No problem.
So all these things are very new to me also. I'm trying to grasp the ideas and things like that. So for me to understand all the concepts better. So do you have any like an analogy from all those to concepts from maybe from a computer vision, more generic tasks?
Yeah.
And give me.
So
yeah.
Yeah. So of course, we publish these methods in journals that are specialized in structural biology, and I spent some time to learn how to explain my work to people that are more biologists rather than computer scientists. And it is very difficult to present in front of a computer science audience. But still, probably the most similar topic in computer science is deformable image alignment or deformable image registration, where you have a template, or you can extract a template in some cases, that contains, represents the general shape of the data. And then you try to match it to every individual data instance. So imagine you have a general shape of a liver, and you have many liver images, MRI or whatever. And then you try to fit this template into every data instance. And by fitting it, you both do rigid body alignment, like you try to physically rotate it, and also to fit it in terms of elastic alignment. You try to fit it into it and see if it has, what is the deformation that should be applied to the reference in order to fit this liver. So this is kind of the most common analogy that I can approach. But of course, in terms of the methods, I don't think normal mode analysis can be analogous to anything, rather than just, if you have a cloud of points and you would like to understand how this cloud of points might deform elastically, you might be able to decompose it into different possible movements that are the least requiring for energy in order to deform. So you'll try to find the easiest path of deformation of a cloud of points, and this is kind of normal mode analysis, which was used in one of the methods. And I would assume that optical flow was obvious, and maybe the way we've used it is different, but yes, as computer scientists, I don't find how difficult it would be to understand that we're finding pixel-to-pixel correspondences. So yeah, that was my sweet spot, let's say, in my PhD, where something actually is easy in computer science is I have applied to the data and got some new method.
Yeah, very, very cool. Thank you for the answer.
You're welcome.
Anyone have any more questions?
I want to ask one more question. You worked on image data, but is there any possibility to add other modalities which can help for understanding or better analysis?
Well, the goal exactly is to try to extract something from the data. So there are different ways, let's say, of finding how macromolecules could actually change their conformations with simulations. So there's this, what is called molecular dynamics simulations, that gives you, you would give an atomic structure, usually coming from different studies, and then it might tell you how it might change its shape. So these X-ray crystallography usually starts from having some proteins in solution that you would crystallize, and then you can image the crystal through some diffraction, and the whole crystal will give you the shape of its individual building block, which is the molecule. And this is how you would usually get these atomic structures. But not everything crystallizes. But let's say you have such an atomic structure, you can apply some physics simulations based on modeling how could two atoms interact in this structure, and then placing it in some environment, saying there are different salts, there's water, and then seeing how the structure might actually evolve over time. And it is a very powerful tool, but it's only based on the atomic structure without any data of really how conformational changes might happen. And for that, you would usually need Cry-EM. And once you have the data, what you can do is to try to combine simulations and data together in order to get accurate representations of these conformations or predictions that match data. And this is something similar that we've done something similar in this normal mode analysis, where we kind of boost the method with some pre-selected movements that are the normal modes. But again, you come to the question of the bias, because you can, if you have a prior on the movement should be similar to that, then if you torture the data enough, it will confess to whatever you want it to confess. So yes, the different modalities could be, yes, the molecular dynamics, matching them with the data. Not thinking now of any other thing. But the main goal of Cry-EM is not to get you only the structure, is to get you the structure from the data without imposing too many restrictions or too many priors. Yeah, but this is always possible, and it is always useful through, though it is somehow, you'll always stay in doubt. Is it the movement, or is it really the movement, or am I finding that simply because I imposed that to my data? So, yeah.
Thank you again for this inspiring talk.
You're welcome. Thank you very much for the invitation again, and I wish everyone luck.
Thank you.
Thank you. Thank you again.
All right then.